Hello, my name is Dagmar Daniel, and I'll be presenting in the Medicine and Health category, and I'll be presenting my project called Spaceflight and the Toxicity of Amyloid Beta Peptides. First, you know the structure and function of the human brain. So the brain consists of nerve cells, which are called neurons, and supporting cells, which are dendrites. And here you can see a representation of the neurons and dendrites. You also need to know about Alzheimer's disease. So Alzheimer's is a degenerative brain disease that is most common early symptom is memory loss. It's currently thought to be incurable, and currently 30 million people in the world have Alzheimer's. And here is a picture of a brain, healthy brain versus a brain with advanced Alzheimer's. And as you can see, the brain with advanced Alzheimer's is severely shrunken compared to the healthy brain. You also need to know about how astronauts' brains fare in space. So NASA is currently doing a study about radiation aging, which is basically showing how astronauts' brains are showing signs similar to aging because of their spaceflight. And there have been several mice studies done that suggest that space radiation may lead to learning and behavioral problems and may resemble aging and even accelerated Alzheimer's. Here is a picture of Alzheimer's underneath a microscope, and Alzheimer's is considered to be caused by amyloid beta plaques. And here you can see is an amyloid plaque underneath the microscope in the brain. And here you can see the schematic representation of the plaque. And these plaques consist of amyloid beta peptides. So amyloid beta peptides are short chains of amino acids which create a large cleavage of a large protein. And these peptides misfolding is what causes them to form plaques, and the structure change is what's causing them to become more toxic. This increased toxicity kills neurons, and there are currently two types of amyloid beta peptides that are known to be the most toxic, which are beta peptides 1 through 42 and 25 to 35. My hypothesis is if the structure of the amyloid beta peptides is changed during the spaceflight, then the toxicity of the peptides will increase. And I'll test my hypothesis by testing the amyloid beta peptides flown into space and on the growth of neuronal cells in a cell culture. So in order to do my project, I found a program called Cubes in Space, which is by Idle Learning, and it's a competition that allows students to design an experiment to fit into a 4x4x4 centimeter cube, and if chosen, it will be flown into a sounding rocket into space by NASA. Here's a diagram of the spaceflight. So here you can see the rocket from the facility, and it launches into space, and at the top you can see the payload separating and descending back towards Earth and into the sea where a ship is sent to collect the payload. My experiment was chosen to fly after the competition on June 21st of 2018 at around 6 a.m. and it was flown at the NASA Wallops facility in Virginia. And here are some actual pictures from the spaceflight that I was able to watch. So for my experiment, I tested 4 peptides. I tested amyloid beta peptides 1 through 42, and then its negative control of 42 to 1, and then beta peptides 25 to 35, and its negative control of 35 to 25. Here is a diagram of the cube itself, and here you can see 4 Eppendorf tubes with the peptides. And after spaceflight, I will be using B35 neuronal cells in a cell culture plate and doing a viability assay. So I needed my peptides and the differentiated neuronal cells and then the triple blue staining solution. So first I differentiated neuronal cells and then treated the neuronal cells with the peptides in triplicate, and then measured the viability of the cells using a triple blue stain test, which stains the dead cells blue. And then I did data analysis afterwards. I used B35 neuroblastoma cells and plated it into a 24 well plate with a density of about 50,000 cells per well. And immediately after, I stimulated them to differentiate them into neurons using 1 millimolar of CAMP. Here are the results for my 1 through 42 beta peptides. And here you can see the peptides plotted for cell viability. And you can see that the negative controls of on Earth 42 to 1, and then the regular peptides on Earth 1 through 42, and then space negative controls 42 to 1, and then space regular 1 through 42. And as you can see, for both negative controls, Earth and space, they both showed cell viability, while the Earth and space peptides that I was testing, you can see both of them were toxic, but the space peptides were even more toxic than the Earth peptides at all concentrations. And here you can see the 25 to 35 amyloid beta peptides. And for this, you can see that the negative controls for Earth and space were both nowhere nearly as toxic as the Earth and space 25 to 35 peptides. However, for the space peptides, they're only more toxic compared to the Earth peptides significantly at the 1 micromolar concentration. The amyloid beta peptides 1 through 42 were more toxic compared to their negative control peptides. However, the space peptides specifically were more toxic compared to the Earth peptides at all concentrations. And then the 25 to 35 peptides are more toxic compared to their negative control peptides. And the space peptides for the 25 to 35 may only be more toxic compared to the neurons that stayed on Earth at the lower concentration. So in conclusion, I could say that the exposure to spaceflight increases the toxicity of the amyloid beta peptides for the 1 through 42 and 25 to 35 beta peptides on the neuronal cells. And the spaceflight may accelerate Alzheimer's disease by increasing the toxicity of the amyloid beta peptides in astronauts' brains. Some caveats would be that I use rat neurons rather than human neurons because human neurons are very difficult to get. However, there have been many studies showing how beta peptides affect rat neurons and human neurons very similarly. And then the effect of the lower concentration at the 25 to 35 beta peptides need to be repeated because the only one concentration showed significant increase in toxicity. And then I also don't know why the space peptides are more toxic. So in the future, I plan to see why the space peptides are more toxic than the HO peptides. Is it either like the misfolding, which I would need help from a biochemist, and then I also need to know what about the spaceflight makes it more toxic. Is it the space radiation or the gravitational forces? So for the space radiation, I could test them using shielding, using polyethylene or aluminum, and flying them into space again. And then for the gravitational forces, I could use a centrifuge to mimic the forces, or using different rockets of different flight profiles, and better yet, using a human carrying rocket that NASA could potentially help with. Here's my bibliography. And lastly, I'd like to acknowledge JSHS, NASA, Cubes in Space, and IB Learning for giving this opportunity to do my experiment and present to you.